Everything's good. Okay, well, excuse me, sorry. All right, thank you for the to the organisers for promoting the abstract to a talk, and thank you really for a great conference overall so far. It's been really good, some really great science discussed. And so I'm going to tell you a little bit about our efforts towards continuous flow hyperpolarization of metabolites via heterogeneous catalysis, sidearm hydrogenation, and membrane dissolution of parahydrogen. Okay, so just a quick summary of what we're going to go through. We'll have an introduction section, just a little bit about the tube-in-tube and sidearm hydrogenation, the construction of our tube-in-tube dissolver, a little bit about the experimental setup, and the preliminary results that we've obtained using our setup. So as we've seen from various talks now, fit sidearm hydrogenation is becoming a robust and convenient way of producing hyperpolarized metabolites. The starting material for this process is typically an ester formed of a target molecule, usually a biomolecule, and an unsaturated sidearm. As you might expect, this molecule is not hyperpolarized, it's not long-lived, and it's not biocompatible. The technique can be broken down into three distinct steps. The first being hydrogenation, where you bring your starting material into contact with parahydrogen, typically done by bubbling with a homogeneous catalyst, and then you produce this hyperpolarized molecule. However, it's not long-lived and it's not biocompatible. And this is where the challenge starts, because these short-lived molecules really need to be either scanned and detected, or the second step needs to happen, which is a polarization transfer. This can be accomplished in usually one of two ways, with either magnetic field cycling or with complicated and ingenious RF pulses. And this produces a hyperpolarized and long-lived molecule, however, still not biocompatible without the last step, which is the cleavage, where you remove the reacted sidearm and you're left with a hyperpolarized, long-lived and biocompatible molecule. And hopefully during this cleavage, you've also removed any of your homogeneous catalyst. And Cavallari and co-workers down in Torino have used this technique with homogeneous catalysis to observe the metabolism of pyruvate in cancer cells, and they produce these lovely spectra that show the build-up and usage of pyruvate in the cells, and also this growth in lactate as well. And we've also seen from a presentation by Dr. Oleg Salnikov that he and Kovtunov and co-workers have also produced carbon-labeled 13C acetate in water that's been hyperpolarized using this same method. And in that talk, we saw they also have done a whole host of molecules and really expanded the library of molecules that can be used. Now, most of the sidearm hydrogenation that's done is done in a batch process, meaning you bubble, you get your hyperpolarized biomolecule, and then you use it. However, I think it would be good, and we saw in Silvio Aimi's talk, that if they could do this in a continuous flow and really just sort of continuously produce these hyperpolarized biomolecules, it cut down on cost and make them easier to implement, I think. One way you can do this continuous flow is using using a porous, gas permeable membrane. And one of the first examples of a tube-in-tube hydrogenation was developed by O'Brien and co-workers as a safer way to perform routine hydrogenation reactions. And this works very simply by having a tube of permeable membrane inside a tube that contains a hydrogen atmosphere. And the idea is that you flow the liquid containing your starting molecule and your catalyst through this tube. The hydrogen dissolves into the liquid and reacts, and out of the other side comes your product molecule. The first use of this with heterogeneous catalysis and NMR defined was by the Kenshin's group in Radboud University, and they used it to investigate the reaction kinetics of using heterogeneous catalysis. You see, you have a simple setup with the tube-in-tube, a syringe pump, catalyst cartridge, and detector. They can do this kinetic modeling of different processes using these heterogeneous catalysis. Now, our tube-in-tube, this is what it looks like. It's composed of four different bits. So we have the FEP tubing, which is the tubing that contains the hydrogen atmosphere, the PFA tubing, which is the liquid tubing. AF2400 is the permeable membrane. It's shown in black here just so you can see it. And also a 316 stainless steel needle, which allows the liquid to pass from the PFA into the AF2400. I've also included the millimeter measurements here for any non-Americans, as I know I struggled with the with going between metric and imperial, which is why I have this nice little table pinned to the wall above my laptop so I can remember what all the various conversions are. So this really works by the process shown here. So we have the needle, which is inserted into the AF2400. This AF2400 with the needle inside it is then placed inside the PFA, and it's all clamped together using the ferrules from a swage lock connection. So as you tighten the nut on the swage lock, the ferrules compress and they pinch down at this point here, encompassing all of the things I've just described, and that seals this against any liquid or gas leaks. In order to perform the reaction, you need a catalyst, and this is where we house our catalyst cartridge in this heater. The heater is formed of a brass cylinder with holes drilled for a thermocouple and a heating element, and also a hole that passes through the whole cylinder for our catalyst cartridge. And it's fed using PEEK tubing, and these are IDEX connectors. Inside the catalyst cartridge is a volume of about 2 milliliters. We have various filters set up. So on the inlet, we have a 10 micron disc filter, some glass wall, then the catalyst bed, which is made up of rhodium on titanium nanorods at 0.2 weight%, and acid washed sand. On the outlet, we have the same filtration system, again, just with the glass wall and a 10 micron disc filter. So what does this setup look like in the lab? This is a bit of a busy slide, but let me take you through it. Hopefully it makes more sense if I had to guide you through it. So first of all, the gas starts over here in the hydrogen cylinder, just a regular one, passes into the liquid nitrogen called Parahydrogen converter, the one that we all know and love, comes out through the mass flow controller into the tubing tube. This is where it dissolves into the liquid, out through a metering valve to the gas outlet, and the metering valve is set at a small flow rate, just so we provide fresh parahydrogen. The liquid is pulled into the syringe from a liquid substrate reservoir via a three-way valve. And then when the syringe has been loaded, it's time to start the experiment. The three-way valve is switched and the liquid is injected, goes into this swage lock T and then into the tubing tube. Once it leaves the tubing tube, it travels to the catalyst cartridge and heater, which is controlled by a control box with a thermocouple. Once it leaves this, as fast as it can, we try and get it into our 400 megahertz variant flow probe with a 60 microliter detection region, out through a back pressure regulator and into the waste collection. I'll point out two distinct things here. The digital pressure sensor allows us to monitor the pressure build up before the catalyst cartridge, as we can have instances where it can burst the tubing tube if the pressure gets too high. So it just allows us to monitor it, and it cuts the syringe pump if the pressure gets too high. And this back pressure regulator here, that allows us to maintain the liquid or have the liquid without any bubbles in it. If you can imagine, without this back pressure regulator, this would be exposed to atmosphere, and because you're pressurizing it, because you're pressurizing the liquid with parahydrogen, this would have the effect of when you open a Coke bottle or something, you just get spontaneous bubble formation anywhere within this line. And as most of you will know, any bubbles in an NMR context are usually bad news for broadening signals, and just can kill any kind of homogeneity you'd have in a magnet. So this is what a typical Altadena experiment looks like for us. So first of all, we inject liquid throughout the whole system, including out of the back pressure regulator. And this serves two purposes. The first is so that we can shim and make sure that everything's okay with the magnet. And second is that we preload the AF2400 with liquid so that the outside pressure of parahydrogen doesn't crush the tube in any way. We use 20 milligrams of the rhodium on titanium nanorods. And we use 100 millimolar of propyl acetate dissolved in deuterated methanol, a flow rate of 1.5 milliliters per minute, and a temperature of 50 degrees Celsius. Once we begin infusion with the syringe pump, we start this pseudo 2D experiment where we take a scan, a 90-degree pulse every 10 seconds. And by sort of the 10th scan here, you can start to see the Altadena signals coming in, and they reach steady state by the 12th or 13th, and then they're just sort of rock solid as they go through all the way up to the 24th scan. By the 4th minute, which is the 24th scan, we stop the syringe pump and then keep requiring to just see what happens, really. Excuse me. And as you can see, this hyperpolarized signal slowly decreases. You might be tempted to think that that's some sort of relaxation process, as I first did. However, it's not that. It's because the pressure of the system is higher than the back pressure regulator, so typically work at 150 to 200 psi. The back pressure regulator is set to 40 psi. That difference in pressure is what causes the it's sort of like a leak, if you like. So the syringe pump is turned off, the pressure has to go somewhere, so it leaks out of the back pressure regulator, which has the effect of slowing down the flow rate, which leads to less signal because you're flowing slower. Sorry. We can extract one of these slices to compare to a thermal to get an idea of the enhancement. The Altadena here is shown in blue, the thermal signal shown in grey. And for HB, we get enhancements of around 60 and polarization levels of around 0.2%. And we've done a little bit of exploration in a couple of parameter spaces by, first of all, varying the temperature from 50 to 80 degrees. Not surprisingly, we get a higher conversion in both cases. We get a higher allyl acetate enhancement, but a bit less of a propyl acetate enhancement. And then we've also varied the flow rate. What's interesting here is that a sort of 25% reduction in catalyst contact time, which is the time it takes the liquid to pass through the catalyst cartridge, results in a more than 50% decrease in the conversion. So there's something that's not quite linear there. And also this 50% in decrease in conversion is made up for in enhancement by a third decrease in transportation time. So the lesson here, I guess, is it doesn't matter how much you make. Really, the main overriding thing is that you get the hyperpolarization to where you need it as fast as possible. So there's as you can probably imagine, there's a bit of further work still to go on this setup. We'd like to explore more temperatures and more flow rates to see if we can get an idea of the kinetics involved in this reaction. Reduction of dead volume between the cartridge and the detection region to allow us to transfer this polarization faster from where we generate it to where we need it. Also, probably need to undergo a redesign of cartridge to reduce the pressures and enable us a wider temperature range. And also the highly non-trivial next steps of polarization transfer and cleavage to form the biomolecule I talked about at the beginning. And with that, I'd like to thank the Bowers Group and now collaborator Professor Hagelin Weaver and the National Science Foundation for the funding. And thank you all for listening.
Thank you so much. That's an amazing, very fantastic talk. And you have I must say, you have a very nice, amazing setup. I just have a question. Do you measure the level of polarization on the carbon or just the hydrogens, right?
Just the hydrogen so far. Yeah, okay. We haven't been able to quite work out the transfer, and we're sort of undergoing a redesign of the system. This is sort of version 0.1, if you will, and so okay. Hopefully version 1 and 2 will be reported at the next PERM. So we just need a little bit of tweaking the system to do with like pressures and I say the cartridge design as well needs to be gotten right.